Welcome already. I think we are almost 80 or almost 90 people joining the first lecture. So as you probably already know, this is a FunDive webinar, and we are starting the new series, like the first series for this year. And today we will have our first lecture that you will hear in a moment. But just for this opening and for very, very short introduction, we just want to say who we are and what we are doing, because we know that among all of the people that registered, there are many people from the outside FunDive community. So we are on the website on the internet, so you can easily find us. It's fun-dive.eu, and you can also scan QR code or like Google us. And we are sharing all information about what we are doing, what is our aim, goals, work packages. We are also sharing the outputs and some records that we are collecting within the project. And also you can get involved. We are launching different campaigns and doing a lot of mycology, fungi related things in Europe, but also we are having a lot of different projects spread around the world, to be honest. So this is more or less the general information, how you can find us and where are the most of the informations. But also, if you are more interested in upcoming events or on whatever events we are organizing or we are involved or what's happening within the mycology, we have in a section called dissemination, sort of like a space on the website where we are posting all of the upcoming events, and we will share information about the webinar there. So if you will like to continue and join us every month for next of the year, you can search for this information over there. And as most of you already did this today, you have to register first before actually joining us online. This is for us and for, let's say, security reason, that we have some sort of control over what is happening. And the procedure will be more or less like that for rest of the lectures. So this registration process will look like that, and then you can join the meeting. Obviously, if you have any problems, any questions, you can text us or like contact us, and we will try to solve the issues. So there was many questions about the recording of the lectures. We will for sure record the lectures if people and the presenting people will agree. And we started the YouTube account, so we are hoping to upload the records there. So we will also share the updates on the shared files and documents and recordings of the meetings and the lectures there. So yeah, who we actually are, the host, it's me, Marta. I'm postdoc in FunDive. I'm working now at the University of Warsaw with Julia Pawłowska, and we are mostly working on sequencing and barcoding of the specimens from the FunDive project. And Glenn is supporting me, or like not supporting, co-hosting and also organizing this. And I will give you, Glenn, voice, so you can now continue. Should I move the slides, or
yeah, maybe.
Yeah, or you will introduce yourself and then I will go on.
That sounds good. So good evening, everybody. I'm Glenn from Belgium. I'm also a mycologist working mostly on deadwood ecology and doing some meta barcoding within the FunDive project. I'll be the technical host for most of these seminars. So if you have any issues, you can find us via the FunDive website, or you can reach out to me at my email address, which I will put somewhere at the event page as well. When you are registering, please always fill out your country code and the other questions that we ask for. You can always provide feedback, which is very welcome. Marta, can you go to the next slide?
Yes.
So thank you for signing up, everybody. We have a lot of participants. We expected at least 100, but we doubled that, and we're close to 250 people registered. So we're very happy to see this large of an engagement tonight. This is just a quick and dirty heat map for all of you to see where everybody is joining from. So we have, of course, a lot of people from the European Union, because the FunDive partners are mostly centered there, but also people from the USA, from China, from India, a few people from Africa and from South America joining as well. So yeah, we're quite happy to see this. And even that Hungary is the most represented country was a nice surprise. And then, of course, Denmark with Jacob's talk. Yeah, next slide, please. So before we go on to Jakob in a minute or five, we'll give the people some time to join. I just want to give you a quick rule first. So whenever we do this webinar series, which will be, like Marta said, once a month or so, we'll always use the same setup. You know where to find us by now. And you'll always be able to join the meeting right away when you have registered. As you will have noticed, your videos are turned off as well as your microphones. I will give everybody video access and audio access at the end of Jacob's talks so that you can ask the questions that you want to ask. In the meantime, if there's anything or if you want to type down a question for later, you can put it into the chat box, and then at the end of the talk, we'll pull all the questions together and we'll have a nice discussion. I think that's the most important things, unless Marta or Jakob want to say something right now. And if not, then we'll wait 4 more minutes and start the lecture. Thank you.
Yeah, I can just quickly say last thing that we are already preparing for the second webinar. If someone would like to be a presenter, like especially from the FunDive, but we are also for now open for people volunteering to give us some lectures. We have some open, let's say, dates for scheduling this. So I will stop sharing the screen. And yeah, we can either wait or Jakob, if you, yeah, I think we have still some minutes, so maybe we can still give a time to the people.
I think we should give people time to join. It's not nice to come at time and then see the things has already started. So let's just wait a few minutes. But of course I can start sharing my screen so that I'm ready with my first slide.
Yeah, I can also, I forgot to say that, yeah, we will probably, because in the end we will have the discussion. And if someone would like to ask a question, you can post them on chat. But I think we will also use this raising hand option, and then we will try to, yeah, read aloud all of the questions that they will be on the record. So.
So do you see my screen now?
Yes.
Yes.
Super. Then we are ready to go. But maybe I'll stop sharing it again so that I can say hello to everybody when I start in face. It's like waiting for the midnight bells on New Year's Eve, but you'll soon be there and I'll get starting.
Here we can start counting from 10 to 1, and then Jakob can.
Yes, on my screen, the time is now 8 o'clock, so Danish time. So I will get started. First, really big thank you to Glenn and Martha for setting this up. It's really well done, and I'm amazed and humbled to see this number of participants joining for this webinar that I will give. It'll be the first in a series of webinars kind of linked to our project FunDive that I will present today. And I will now start sharing my screen. And just to make it clear, my intention is to talk for roughly 45 minutes and then having time for questions. So as already pointed out by Martha and Glenn, you can put questions in the chat or write them down and use the raise hands signal in the end of the presentation when we are ready for that. I guess you now see my screen. Is that correct? And we'll get started. So today I'm gonna present this project that we call FunDive. And my presentation will have three main parts. First, I will provide a little bit of background. And then, since this is the first seminar in the series, I will spend quite some time giving an overview of the project. And then I will give just some very first results from the project to give you a little bit more than just talking about what we intend to do. So stay tuned for having some first insights, including unpublished results that we are still working at. So this travel towards relevance, this background part is partly personal, but it's also giving insights into how the field of fungal conservation has developed. Back in the 1990s, I was involved there was a plan to put fungi on the Berne Convention, making them relevant for also the EU Habitat Directive. That was actually a very professional work, suggesting 33 fungi to be added to the Habitat Directive, meaning that they would have consequences for conservation in Europe. The suggestion was turned down, and a kind of still appreciation of the work that was done. A number of people, including me, was hired to do this report for the EU on how to conserve macrofungi in Europe, and that led to also some papers about fungal conservation, including a status of conservation of fungi in Europe and future needs, giving maps of where we had checklist of fungi and how was the actual conservation status. I also did a perspective paper in conservation biology with several co-authors based on a symposium at the European Conference of Conservation Biology, kind of making the case that fungi should be relevant. And a lot of other things has happened. This is just my personal part of it. Then in 2009, I got involved in the Danish Fungal Atlas, involving citizen scientists in mapping fungal diversity and making that information available for nature management in Denmark. And based on that project, I also engaged in other projects with colleagues from other countries on how we could work with a more integrated approach to monitoring fungal biodiversity, combining sporocarp surveys, eDNA surveys, and so on. I got involved in European collaborations based on the Danish Fungal Atlas on bringing data together from different projects led by Heilmann-Clausen from Oslo and Carrie Andrew as the postdoc doing all the hard work, working to get fungal data on the map. And I was also involved in a project where we compared eDNA, this meta-barcoding approach to fungal diversity, with sporocarp surveys, trying to investigate how they kind of provide similar or complementary insights to monitoring. And then, I mean, things has just developed since then. Fungi has really, since I was undergraduate student in the 1990s, and fungi were considered like a little bit on the sideline. I think it's fair to say that fungi have now entered a center part of the scene, maybe still a little bit as a coming up group, but there's a lot of emphasis. And fungi are sometimes even seen as the organism group that can save the world, but is probably a little bit of an overstatement. But at least there's a lot of emphasis on fungi. The word fungal is now getting more and more accepted. And we also have now fungi on the international red list, meaning that fungi are moving from being a neglected group of odd plants in the sideline of conservation to be really considered something in their own right. And it's important to state that still we have a challenge, because the fungal kingdom is huge, and there's a tendency that that people that are not really working with fungi expect that you as a mycologist know everything about fungi. But it's I think it's not always, even by mycologists, really recognized that we are working with a super complex kingdom with a lot of undiscovered diversity. Even within the Basidiomycota, there's a lot of unknown diversity. And then there's all the other fungi. There's all the Ascomycota, which include quite many macrofungi, but also many microfungi. But that's where we have the biggest diversity. And with some of the tools we're using, we can basically explore it all. And sometimes people kind of look at fungi as something you can just study in one go. But I think no zoologist or botanist would consider publishing a study that is including both birds, nematodes, and mammals and reptiles in one study to say, oh, now we have studied the zoological diversity. When we do the fungi, we actually do more or less like that. So I think it's fair sometimes to say it's okay to zoom in on smaller groups of fungi, and also when we do the whole thing, really recognizing that this is huge, it's complex, and it's covering groups with very different ecological strategies and organism size and all that stuff. So that was just one of my favorite points to say when I start talking about fungi, that it's complex, it's huge, and it's a lot of different organisms we are working with. So just to give a little bit insight into the monitoring methods, with the kind of things we are looking at. When I was a student in the 90s, you could do some molecular work, but mainly you could take one sample and get one species ID out of doing the sequencing work if you were lucky. But since the last 20 years, it's been possible with metabarcoding to basically explore the full fungal community from just a soil sample or from air or from sawdust from wood. At the same time, citizen science has really moved forward as at least something we talk about. I mean, the number of observations in GBIF, as shown here, is really growing. The number of papers dealing with fungi and citizen science are growing. But in reality, it's an old tradition, just showing two of the most famous Danish mycologists describing a lot of agarics 100 years ago. They were both basically teachers that had mycology as a side business. So today you would probably call them citizen scientists. So it's basically new words for something that has more or less been the tradition within mycology for a long time. But of course, there's many new ways to do it, especially facilitated by the fact that we have all these molecular tools. IT tools, online databases that can be used. So, so this is all the background, my own history of working with fungi, the overall development for fungal conservation, and the strong development in approaches to study fungi mycology. that was basically the background for applying the money and in the end getting the FunDive project funding. And FunDive has one overall goal, and that is to put fungi on the conservation map. And then we have some underlying goals that I will not go too much in detail with here, but you can read them and take more time to look at it carefully afterwards. The funding is from the Biodiversity Plus joint research call on improved transnational monitoring of biodiversity for science and society, where we are basically dealing with all the 3 themes that were specified when they gave this call, the Biodiversity Plus, the innovation and harmonization of methods for managing and collecting biodiversity data, addressing knowledge gaps on the biodiversity status and trends in biodiversity data, and also looking at what data is actually available and can be used already for looking into these questions. And to put our project in a nutshell, we are basically looking at 2 key ways to study fungal diversity. One is to use standardized metabarcoding or eDNA techniques, and the other one is to really involve citizen scientists in the work. In the citizen science part, we work with projects or missions to engage citizen scientists in projects across Europe. And then we also work with making apps that can help in species identification based on AI. In the eDNA part, we have a common project across Europe focusing on pine forest. And then we are working on how these 2 different types of data come together, because there's issues here. Citizen scientists looking at fungal spore carps are dealing with morphological species concepts, whereas the OTU list that you get out of metabarcoding is based on molecular species IDs, and they don't always fit. So sometimes a species that is super common in the eDNA data cannot be adequately named because there's issues with the reference databases linking the classical morphological species concepts with the eDNA data. So to facilitate and bring a stronger platform for doing that work, we are also having a mission or project on DNA barcoding and annotation to improve the databases so that it's at least getting more easy to compare these 2 different types of data. And finally, we want to bring this data, both the new data we are generating and the existing data, into exploring fungal biodiversity patterns in time and space, evaluate the conservation status, and basically bring this knowledge into the scene of conservation in Europe. More officially, it's structured like this. I will not go in detail with that, but that's how these different tasks are developed into what we call work packages. And in total, we have a funding or a budget equaling 4.4 million euro. It's distributed across 26 partners, so the budget per partner is not super big, but still there's many partners involved in each work package. So we do have some funding to work with, and the project is running from 2024 and into ending next year. As I mentioned, we started out with 26 founding partners in 16 countries, but we building on the fungal metaphor, the mycelia, we have expanded further since then. So we have quite a high number of affiliated partners, and this list of logos is not even complete. So, but anyway, we now have at least 24 countries in Europe involved in what we're doing. And it's been really a pleasure to see how people have engaged, including people that had to chip in with their own time and own funding. That's been really amazing to see, and something I really appreciate a lot as project coordinator. Yeah, maybe I should have introduced myself as the project coordinator of FunDive, but I think that is clear that that's my role in this project. So to present the project in slightly more detail, I will go through the work packages in more or less the same order as they were presented in this FunDive in a nutshell slide. So not starting with work package 1, but in this case with work package 3, which is the citizen science work package that is basically about engaging with what we call volunteer mycology. So it's not only kind of the really untrained citizen scientist that just wants to learn a bit about fungi, but it's also including the really highly skilled amateur mycologists that are deeply already engaged in mycological associations and so on. So we are working with a full range of engagement with people that do mycology without being paid for it. And it's led by Julia Pawlowska and Danny Haller as sub-coordinator of this work package. We run different projects. Currently, we have 16 projects running, and some examples because we of course try to investigate how this works and how we can engage people in a way that makes sense for people. So we have 2 campaigns, or actually 3 campaigns on gastromycetes that are in the process of being assessed as a group for the IUCN Red List. So we have campaigns on Gastrom and Tulostoma with that specific focus. We have other projects that are focusing on specific genera or other groups of fungi that are where we want to understand the diversity better, and that's including even lichens like dermatocarbon. It's including microfungi like Hesperomyces that are those small Labulomycetes that grows on beetles, and also macrofungi like Paxillus. We have a project on pine forest fungi that is kind of linked to what I will present a little bit later on the metabarcoding, seeing how different methods compare when it comes to mapping fungal diversity within a specific ecosystem. Then we have some bigger and more inclusive campaigns that are maybe more classical citizen science, where many people can participate without having too much experience with fungi. So one on alien fungi, where we try to see how widespread non-European selected fungi are, and one on urban mushrooms that will not really running this year, but will run more intensively next year. And finally, we have a more specialized project that I will present a little bit more detail later on making types for fungi, missing those. I'll present that later. So quite many different projects with different scopes and requiring different levels of engagement and expertise to contribute to. And contribution is basically pretty simple. We have on our homepage, we have these guides on how to contribute that is presenting pretty detailed information on some of the groups. We have collaboration with the Pluto F Go in Estonia, but we also accept records made on other recording platforms. You can read more about that on our homepage. But if you use the Pluto F Go, the records are going automatically to the online map we have on the homepage, and then it's directly linked to the sequencing process that, at least for some of these groups, is crucial. So and it's all coordinated by national contact points. So it's also something you can read on the homepage. But basically, if you go to see this Get Involved page on our homepage, you can find more details. And if you are representing a country that is not yet involved, this can be organized. We just need a national contact point that can handle the kind of national level coordination of the efforts, so that this is serving as a link between the overall project coordination and making sure that that there's some filters, and also that the Nagoya protocol is handled in an appropriate way. But reach out if you want to know more, or check as a first step our homepage for how to contribute to this. As part of Work Package 3, there's also campaigns to, so some of it is, quite a lot of these campaigns are involving sequencing with nanopore, mainly coordinated in Poland, but in some countries, including Denmark and also, also I think Hungary, the sequencing is actually done at the national level, and then data is compiled in centrally afterwards. But we also have had some campaigns to bring out the molecular labs for the engaged citizen scientists, having hands-on experience with using these new platforms that are making it at least more approachable for amateur mycologists or volunteer mycologists to actually sequence their specimens. So that's something we are starting up at, and especially in Poland, has been really active with developing this. I think there's still some way to go before this is being the same thing as working with a microscope, but we are going in that direction, so that that barcoding specimens is becoming something that is approachable for volunteer mycologists without a big budget. Then we have the Work Package 2 that is about developing machine learning tools to help people identify their fungi. It's led by Lukas Pisek from the University of West Bohemia, and it's really the smallest our work package is. There's not too many people involved in that, and it's mainly to develop things that we already have running in the Danish fungal atlas, but to develop it further and bring it out to a bigger audience via the Pluto F Go app. So it's they have developed the AI that is now helping people to get closer to identification of fungi. I mean, when I first heard about this idea, I think it was crazy and it wouldn't work, and it would also take out the kind of challenge in working as a field mycologist. But after working with this system in Denmark for now 6 years, I have to say that I'm really impressed of how it performs and how it also works as having somebody close to you that actually knows the fungi. You also see all the challenges that some people take it too literal, because this is just a tool that can help you identify your fungi. You have still to accept that you are much smarter than the AI, but still it's a very powerful tool that also helps me to get an idea of what I have found when I sometimes find fungi that are outside my comfort zone. So that's a tool that I now embrace very much. Work Package 4 is then the other big sampling campaign but this time using eDNA and metabarcoding for investigating fungal communities. It's coordinated by André Gross and Håvard Kauserud from Switzerland and Norway. And we developed a joint project on pine forests in Europe. Here's the sampling protocol. So we compare. First of all, we of course look at one system across Europe, but we also compare how different substrates contribute to understanding the fungal diversity in this ecosystem. So at least in some of the plots, we are sampling both dead wood, soil, and air. So we have these passive spore traps that are presented in this paper that are simply collecting dust out there, including spores, and it is then giving insights in which fungi are actually active in producing spores in the field season. And finally, we also did, at least in some of the plots, joint sequencing of dried sporocarps from the whole plot, as a kind of not really eDNA, but a complex sample of many different fungi, as a test of using this as a rapid way to make community ecology based on the sporocarps. So combine all these different methods in a lot of plots. Our main focus is Pinus silvestris, where we have a total of roughly 120 plots, including 46 intensive plots. That's where all the sampling is done, including the sporocarps and the spores from the spore traps, and also with several sampling occasions for the spores and the sporocarps. And then we have 74 opportunistic plots where only the soil and the wood is sampled, so no recording of sporocarps. And as you can see, we are covering more or less the full extent of pine forest in Europe, excluding Russia and Great Britain. That is not part of the funding scheme in Biodiversity Plus, unfortunately. And of course, there's also some gaps in the Balkans and Turkey. And we have a smaller project on Pinus sembra in the Alps. And on top of that, Frank Ricard from Montpellier has coordinated a side project on fungal communities in the Pinus nigra complex that is basically distributed across 5 different subspecies of Pinus nigra that we have sampled both within and outside their native ranges, with a total of 61 plots. So basically, and as you can see, that's even extending to northern Africa and Cyprus. So basically, we are covering pine forest fungal diversity across all of Europe, across 3 different key species within the genus. We don't have any results on this yet. The metabarcoding results are just being analyzed now with the bioinformatics. So within a few months, we will start to have data to work on to see how fungal diversity patterns are then reflecting biogeography and other drivers. The next work package I'll present is the work package 1. That's the one that is basically working to improve the molecular backbones for annotating fungal samples that are sequenced. It's led by Jorinde Nuiteng from the Netherlands, from the Naturalis Biodiversity Center. And it's mainly focusing on solving issues in relating to having more clear molecular identities, as mentioned. And just to give you one example of how deep the problems are, we can take this violet coral, the Clavaria solengeri. That is basically, this is information from the IUCN Red List. So it was one of those species assessed globally to be vulnerable, with a distribution map covering most of Europe. If you go into the main platform for fungal eDNA data, Unite, and that is the last, it's not the current version of the so-called species hypothesis system. You can see that there's 9 different species hypotheses, different at the 1.5 level in the ITS region of the DNA. So it's clear that Clavaria solengeri seems to be more than one species. And which one is then the one that is red listed and the one you will record as a sporocarp in Europe? That is the big question. Here's one example of how one of these OTUs look. And when you then go to Index Fungorum and look into the details of Clavaria solengeri, you will realize that this species was not described from Europe. It was described from Indonesia, from the island of Java. So there's really something to disentangle here, and sequencing appropriate types and reference sequences from the field are really important to understand what's really going on. In this particular case, the issue is actually solved. So there was a paper coming out last year that was actually initiated much before FunDive that actually realized, oh, there's something wrong here. Clavaria solingere is the species from Indonesia that is not the same that is redlisted in Europe, and now we have new names for the 2 different species that occur in Europe. So that's kind of the work we want to do. We want to disentangle these complexes and link the morphological and the molecular species concepts. And to do that, we have mapped how many types are present in major collections in Denmark, and then we will try to sequence a fair share of these. We don't know yet how many we'll reach, but we are still working to at least reach the, maybe not the 10,000s, but at least the thousands of types sequenced across Europe. That's the goal. And this is about, of course, also developing good protocols for old and degraded material. That's a big task within that work package to find methods that can be used for 200 years old collections. That's not always simple. It's also about annotating the databases, primarily in UNITE, so that the so-called species hypotheses are more directly linked to what we really accept as good species. There's a lot of, I realized that this is a bigger task than I thought in the beginning, but we still have a high ambition to bring molecular identification forward. In some cases, especially for the classical taxa, there's no types that can be sequenced. And that's a big issue. I mean, if you take one of the most common Russula species in Europe, Russula vesca, that you can find in every fungal book as a very easily identifiable species, and then you go into to try to find it in papers based on metabarcoding, you realize that it's never recorded because it's not really linked to a clear species hypothesis, or if it's done, it's done very recently. So if you have some metabarcoding data, you cannot find Russula vesca. And then in some papers, you can see some names described recently from China being some of the most common Russula species in Europe. And to handle that, it's not enough to go to the collections, because for many of the old names, there is no type that can be sequenced. So for solving that issue, we are designing campaigns involving citizen scientists to find new so-called epitypes or neotypes, depending on the exact conditions, that can be sequenced and fix the way that we use names for these classical fungi. And that started out last year with a Danish test case where we focused on 16 fungal taxa described from Denmark, and we are getting some potential types already. For instance, this Flammulasta granulosus that was described by the earlier presented Jakob E. Lange. He never, only very rarely conserved any material, so we don't have anything to sequence. But in this case, we found a collection on the same island where Jakob E. Lange worked. And as you can see, it seems to be a very good fit for the type. So probably this will be designed as the epitype in this case, and be sequenced and deposited as a reference in the UNITE system. And this year we are continuing with last year we launched 20 campaigns for taxa across Europe. It's one of the more demanding projects we have realized in order to get people involved, but it's something we want to develop into the future because we see a big need to collaborate across Europe to settle these issues. Finally, work package 5 is about exploring fungal biodiversity patterns based on already existing data, led by Franz Sebastian Krah from Bayreuth in Germany. And dealing with, I will present some results from that project, but it's basically a lot about looking at spatiotemporal patterns, looking at a very important question, seeing how eDNA versus sporocarp data is providing similar or contrasting patterns of fungal biodiversity, and also how this is contributing to knowledge about conservation status for these fungi. But I'll present some results from this work package that gives more insights to what we are currently doing. So that was the presentation of the overall project, and then I will use the last 10 minutes to give you some first insights into what we have learned so far. So we are still rather early in the process. Data is getting ready. We have the first papers coming out. We have posters presenting at the last International Mycological Congress. But the 2 next years, we will see a lot of things coming out when we really start working with the data. So this is really an early insight into what we're doing. In work package 1, as I mentioned, one of the things is to sequence old types. And here's one case that we already did very early in the process, where we took 96 types from French collections that failed in Sanger sequencing in the first go. And then we tested out here in Copenhagen the Illumina platform that is better at capturing small fragments of DNA and can also capture not only the target in Sanger sequencing. You will normally only get the most abundant DNA out, but within Illumina, you will basically get all varieties of the target region in the DNA out, in this case, ITS2 region. And you can see for this case with almost 96 collections, we actually were able to get a useful DNA out of more than half, distributed across quite many genera. And in 17 cases, we could consolidate old names or reestablish them as the most relevant names for taxa that missed annotation in the Unite data platform. In 2 cases, we could provide an earlier name for already existing species hypothesis with a name. And in 31 cases, in most of the cases, actually, we could say, oh, this more recent name published is basically a synonym of something already published earlier. So it's not a new name, but just clarifying that it's not a separate species, which is just equally important. So just to take one of the names that we could consolidate was Cotenaius ulvaceus fuscus, that is now more clearly settled and linked to a type sequence, enhancing the future annotation of all sorts of fungal DNA data. In work package 3, we have the first paper out about mapping the landscape of mycological organizations in Europe. You can see here in the first graph how the number of mycological association has risen since the first one was established around 1860 to now being more than 70 across Europe. And that also means that some countries, and that's very clear from this paper, that in some countries like France and Italy, to some extent Spain, and there's a strong tradition to have really regional mycological societies, where other countries like, for instance, Denmark and Sweden has only one mycological society. So there's big differences in traditions across Europe. That's also clear when you look at the organization level. So this is basically the proportion of people out of the full population that is engaged. You can see Norway, Switzerland, and Slovenia has the highest organization level of all countries in Europe, whereas it's really low in Belarus and Serbia. So different traditions, and also different traditions in who is engaging in these associations. Most are in this dark blue category, where it's including mainly non-academic volunteer mycologists, and then you have those that are exclusively non-academic members, and also a few that are only academic or mainly academic members. So there's quite a difference across Europe and also within some countries in how mycologists are organized. We're also starting to get the first insights into to some of the campaigns we have done, where we have done the barcoding of collected material. And the flagship project is the one on gastrum that is, as I said earlier, intended to provide a stronger background for red list assessment of this genus, basically at the global scale. So in this project, we have sampled roughly 260 specimens that have been sequenced from 22 countries. And of course, there's many new country records that are kind of expanding the known distribution of several taxa. We have roughly 40 taxa detected in total, including roughly 7 that seems to be new to science. So even within Europe, a campaign like this one, running only for 1.5 years, has provided important new insights. And of course, when you start doing this, you also realize that fungal taxonomy is sometimes burdensome. So one of these undescribed species seems to be this one, that is basically presented in a recent paper on the genus Gastrum as Gastrum saccatum, but it's pretty clear that it's not the real Gastrum saccatum, because that was described from Brazil and is placed up here in the violaceous part of the tree, whereas these 3 samples represent a taxa that seems to be present only in Europe and seems to be lacking a name so far. So interesting results, but still some work to be done before it can really be effectuated into updated and completely updated status of gastrum. At least that's something that will take some time. In work package 5, we have a paper out in as a preprint led by Franz Sebastian Krah on changes in fungal alpha diversity in Europe over time, based on 5 million records. And it's mainly showing that, I mean, in the red regions here, there seems to be some negative change in alpha diversity in fungi, whereas the blue region seems to be regions with increasing diversity. And it seems that this is partly related to nitrogen deposition that seems to have a negative effect on the diversity of that's down here, especially in the change model here. We can see that change in diversity is linked to nitrogen deposition, whereas regions increases in diversity seems to be related to regions that is getting wetter, warmer and not drier. So it seems that climate change is generally increasing fungal diversity in most of the parts of Europe we have been investigating. Then Ovidio Copot, that is doing a postdoc here in Copenhagen, is working hard on looking at the conservation status of fungi in Europe. And here you can see again the map of fungal records that we have available. See that it's very uneven. This is a log scale. So some countries have a lot more data than others. And we can also see that the quality of the data is very uneven. So if we only if we exclude data that has less precision than 1 kilometer, you can see that most of Germany, big parts of France and Spain and Iceland are getting really, really dark. That means that we cannot use the records with high precision. So that's been a really surprising and stark realization that the way that we record fungal diversity in different countries has a huge impact on how precise the data is. And when you want to see whether rare fungi are recorded within or outside protected areas, that's an important issue. So for this really strategic conservation planning, I think some countries could maybe learn to record fungal data in more smart ways than they do now. Then also, and this is really work in progress, so that might change in the end, but we try to look at what countries have a very good coverage of protected, red listed or species at risk within protected areas and which do not. And a little bit to my surprise, it seems that Denmark is one of the countries with the best coverage of species at risk within protected areas, whereas based on the current analysis, Norway, Switzerland, and partly also Sweden is not super good at protecting their red-listed fungi currently. Also looked into how the conservation status and the ecological strategy affect conservation protection level, and at least we can be happy to see that of the critically endangered species, so that's the most high threat category before you go really extinct. For those, the protection level is pretty high, more than 75% in areas are within protected areas, where it's lower for the lower threat categories and also for the species that are considered least concern. And then it seems also that wood saprotrophs, BS here, are in areas better protected than, for instance, the ectomycorrhizal fungi, which is interesting and give some insight into how the protection of areas that is mainly not based on fungi is actually considering fungal diversity. Also see differences between countries, where there's again big differences, again with Denmark, the Czech Republic, and Lithuania as basically the three countries having the highest number of, at least the known records of red-listed species protected, whereas Norway, Switzerland, and also Iceland seem to have a very low protection level. So that was basically my last slide with contents, and I just have to say a really warm thank to all my partners in FunDive. Without you, I wouldn't have been able to present anything interesting today, so it's great to work with this amazing team of colleagues. And finally, I just want to say thanks to the audience for your attention, and now it's time to ask questions. So thanks.
Yeah, thank you very much, Jakob. It was very, very nice. And also thank you for mentioning that I forgot in the opening that you are actually the main, let's say, leader of the entire project. So thank you again. And I think we can start with the questions if there are any questions here. I see that there is a one in the comments, so maybe I can read it and then Jakob can answer. It's from Erin. Have there already been surprises from Indian sampling? Example, species which are rarely recorded within classical field mapping being more commonly found through eDNA?
Yeah, unfortunately, we don't have the eDNA results yet. So I cannot answer that question, but I mean, we have been looking into existing data that are present, and it seems that there is many surprises. I can recommend one of the papers from Ovidio's PhD project from Estonia that looked very carefully into this aspect, but from FunDive, we don't really have results yet. Unfortunately, we will have it within a few months. We will be able to say something about that.
Okay, so if there are more questions. Oh, there is. If you want to say your question aloud, you can also do it. It's just for us that we will. If you don't want to do it by yourself, we will read it, and we want to keep the questions and the answers on the record. So there is another one from Lisandra. So I will also let myself to read it. I heard you mention Unite, GBIF, and EUCN Red List. Are there any other public or open sources databases, reference repositories, or functional annotation systems where we can both contribute field data, sequencing, identify taxa, etc., and search for insight related to the taxa we are seeing in the ecosystem we are working in.
Wow, that is a big question, and yes, there is. I mean, and it's I generally hate when presenters just provide a lot of abbreviations of different associations, as I actually did. So I hope most people are familiar with IUCN, GBIF, and UNITE. And if that's not the case, I'll be happy to take that afterwards. It's probably not where to start. I mean, GBIF is the big hub for all sorts of open source biodiversity data. So it's taking in data from a lot of other sources, including iNaturalist, including many national recording schemes. It's also linked to UNITE, that as I already mentioned, and also to other platforms. So GBIF is kind of a hub for other databases that want to share their data. And I mean, in FunDive, we have data also from Germany and France that is not yet shared with GBIF, but where we have collaborators that can help us with that data. And it's one of the goals is to, I mean, there's different traditions in different countries, but we generally, I mean, my philosophy is also that data should be as free as possible. We also in this project adhere to the FAIR principles, that data should be findable, accessible, and reusable. I can't remember what the I is for, but at least that data should be open and accessible. But of course, we don't have control of how national recording platforms are doing their job. So for citizen scientists, it depends on your country of home where to do it best. And then for the eDNA data, that's a different story there. We are also counting on several different databases, or at least two different databases, UNITE and the global fungi database. And also GBIF has eDNA data.
Thank you, Jakob. We also got some nice recommendation of the conference, and I think as far as I remember, FunDive will go to this European Citizen Science Association conference this year in Oulu. But yeah, it's in the chat if someone would like to know more details. We have one more question from Matthias this time. I assume Matthias from Canada, so hello. What are your main arguments for securing funding and grants to support these projects? Europe seems to be one of the leading regions in the advancing fungal conservation. For other parts of the world where the importance of fungi is still largely overlooked, that approaches have proven most effective to convince governmental authorities, funding agency, and institutional partners. So what would be our advice, I guess?
Yeah, that's a good one. I mean, the main arguments we use for this application, and I'll be happy to share it with you if you send me an email, and I can share the kind of the main argumentation to the biodiversity for getting funding. But I mean, we make this point that fungi are fundamental to ecosystem functioning in all terrestrial ecosystems. So neglecting fungi is a big problem. But fungi has been neglected in conservation. But now, thanks to metabarcoding and thanks to improved collaboration with citizen scientists, we actually have tools to bring fungi into the scene. So that has been like our overall kind of argumentation for doing a difference, that we really have something important to deliver, not only for fungi, but also for understanding ecosystems more broadly with the key functions fungi perform.
Okay, thank you. Any more questions? I can't see any new question in the chat. But yeah, you can always contact us. We have contacts on the website, and also we are, I guess, almost everywhere in social media. Like we are more I would say, visible on Facebook and I think Blue Sky for now. So except of our website, so we can always like find some contacts and contact us.
Yeah, Marta, since you talked about the Citizen Science Conference, I just want to advocate for another conference coming up even sooner, which is this conference organized by SLU in Sweden on fungal conservation in Europe from evidence to action that will run from the 14th to 17th April in this spring. So I think that will be a really go-to event for everybody interested in these aspects and able to join. I think the program looks really interesting, and it's both a combination of hand-on workshops and presentations and workshops. So I can highly recommend that one. I can put it in the chat maybe.
Yes, that would be great. Okay, so I don't know if we do have any more questions, but if not, then I think I can say again thanks to Jakob. Thank you very much for your first lecture and introducing to the whole project. And as we said, we will be updating about upcoming meetings, and if you will have any advices or ideas or whatever comments maybe on the time. I mean, we decided that it would be better to keep them in the evenings, that everyone can join also after work, but we will be like open for some discussions. Oh, I see that there is a question. Are there any plans to organize a conference meeting or other event in Belgium? I work on the French-speaking side, focusing in particular on harvesting and identification in nature parks, and I would be interested in meeting with you as a speaker. So I know we have a lot of Belgian people here, but also someone else that not only Jakob can answer, but yeah, you can.
Yeah, I think I might be able to answer this. There's not a direct FunDive meeting in Belgium that's planned, but if you leave your email address, then I will reach out to you. And I thank you for at least offering up to present the talk.
I see there's also a hand from Jeffrey.
Yes. By the way, everybody who wants to share their video or wants to unmute themselves when they ask a question, please don't all start speaking. They have the opportunity to do so by now. So Jeffrey, I don't know if you want to.
Yeah. I'll just
Thank you.
share the audio. You don't need to see my couch at home, but thank you for the talk, Jakob. That was very good. I was just curious if you had an opinion, since this project is generating so much new sequence data, including a lot of citizen science sequence data. And right now we have, Pluto F, which feeds into Unite. We have GenBank, we have Global Fungi, and now we have iNaturalist, which is over 100,000 ITS sequences. Do you see these as complementary? And it's nice we have some non-overlap and some overlap, or is this just a challenge when examining the data to figure out what's unique and what needs to be cleaned? Like, is it good to have this, what's tending towards kind of a decentralized system, or is it better to have it all together? Do you have any thoughts about these sequence data?
I mean, that's a very good question, and I mean, in an ideal world, everything would be linked and combined together, I mean, that's what I would love to see, and I would love to see things convene towards one unified system. I don't think it's smart to have several different systems that are not linked. I know that there's efforts to bring these things together, and then there's, of course, the really big issue about technologies for link. I mean, the whole bioinformatic technologies, including AI tools to bring species concepts better together. And I mean, also on the sequencing side, we'll start to have access to maybe not full genome sequencing, but at least full genome skimming that will provide much better resolution in taxonomic issues. So, I mean, there's a reality and then there's the ideal situation. And I think I would love to see things come together, but I also see a reality where I think most people that works on these processes wants to bring it together, but it's not always super easy. And some systems are starting to get a little bit old in the way that they work. But I'm all in for data sharing and also for open data annotation. I mean, that's one of the problem with some of the older systems that are super heavy to annotate. And what I have learned from working with the Danish Fungal Atlas is that the more easy you make the annotation, the more efficient is your progress in making tools that are truly useful for the end users. But yeah, I also see the challenges in actually doing that.
Thank you.
I see one more raised hand from Paula Ortega.
Yes, hi. Thank you for the talk. I had a question about eDNA in soil often shows a lot of fungal species and different and a big amount of them, but they are often not described. So how do you think, or how to describe species that we know that they are dead, but they are maybe not so easy to cultivate or to define? So via genome-based description, or what is like the next step in those species to be able to be into accountability in the decision making of nature conservation?
Yeah, it's not something we are directly addressing in FunDive. I mean, we are not really going into this interesting question that I mean is discussed a lot and was most recently discussed in a big meeting in the Netherlands that I didn't participate in. So I'm not completely updated, and I don't have a strong standing on this exact question. But what I would say is that mainly data gets poorer and poorer over time. But when you have the raw metabarcoding data with all the sequences, even if you don't have a species to match it with now, the chance that you will be able to match many more of your unknown OTUs in 10 years is growing. So I think it's very important, and that's also something GBIF is working on, is to facilitate that metabarcoding data is kept for the future, because the number of unknown OTUs that you will be able to identify in the future, irrespective of the way that fungi are described, whether we will go on and describe species just based on sequence information. The chance that we can understand this data better in the future is big.
Okay, thank you very much.
Okay, thank you. I see one more question on chat from Erin. Will there be more project upcoming this year, or will the focus remain on current projects? I don't know if it's only on campaigns or all projects within FunDive, but yeah, Jakob, you can.
Yeah, at least we are going into the last year in 2026 where all partners have a budget. In 2027 is basically a project extension that is reflecting that we were a little bit delayed in the project start due to funding issues in the biodiversity program. So we don't intend to start new projects at the moment, but we do have other applications out, including a EU cost action that might make it possible for us to extend our activities. But otherwise, I don't think we'll start new projects, except that we will probably put some more target species for the typification project. But otherwise, and also this urban fungi, urban mushroom campaign will be more kind of focus at in the coming year.
There's also, I think we will keep updating people on the website with current campaigns. We have this event section, and then every time there's something happening, is posted there. There is also a question related to, I think, Matthias' question, but this time from Saidi. First of all, I would like to congratulate you on your talk, and the question relates to this project. In the event of collaboration with Africa, and more specifically with Algerian naturalists, what forms of support, knowledge exchange, or recognition of local expertise could be envisioned within such a partnership?
Oh yeah, very good question, and something that I mean, we grow as a mycelium, and we also learn as a mycelium, and the more we extend beyond our the first partners, the more we also realize that we have to handle various issues. I mean, for this academic side of it, we have some guidelines on how we appreciate courses and stuff like that. But working with citizen scientists and naturalists is making things even more complex, and we are in the process of looking into how we do this. So it's something we are trying to find even better principles for in the future. But generally speaking, we of course adhere to, for this academic publication, to the Montreal Protocol and other similar protocols, and then for the more kind of more informal appreciation, I think I think that the main quality of being engaged is the chance to collaborate with people that have the same interest for you. So it's kind of based on joint interest and working together. And of course, our resources are not they are limited. So moving beyond Europe is not something we have really considered so far. But please reach out to me, and if you want somehow to engage. I mean, the collaboration with Algeria that we already done was done by one-to-one collaborations with Frank Picard and collaborators. But we don't have a policy for more generally moving across Europe, and I don't think we have the budget to really handle that at the moment, unfortunately.
Okay, thank you. I see one more question. Do we still not have too much primer bias to be able to catch all the fungal diversity in eDNA samples? Which one with one primer set? That's a question.
Yeah. No, I mean that's of course how it is, and I mean, what we do is biased, even with beta barcoding, and that's I think even with more than one marker or one primer set, it would probably still be biased for many different reasons. So we don't argue that we have a complete sampling of fungal communities, but at least we do it in the same way across our study systems to make it so that we're at least looking at the same thing across the scale we're looking at. But we are not really, it's not a question we do have a little test case with long versus short reads and different sequencing platforms, but it's we are not really making tests of different primer sets and stuff like that.
Okay, thank you. There's one more. One main criticism I'm receiving when advertising fungal conservation is that fungal protection is achieved through habitat protection, and fungal habitats are already covered by animals and plants that inhabit them. Could you name some specific cases, practical examples to deconstruct these lines of arguing? That's a tricky, I would say. I would not.
Yeah, I mean,
Yeah.
we are working on this manuscript based on the analysis. We are not there yet, but I think when we make this case, we will also emphasize the cases where specific fungi, specific ecosystems that are or habitat types that are important for fungi that are clearly not covered adequately based on the current way that conservation is planned. I would like to add to that my perspective on conservation in general is that it should be system-based rather than species-based. So I'm really not a big fan of saying, oh, now we also need to do a lot of fungal-based conservation. I would rather say that we should learn how current conservation gaps are linked to important aspects of ecosystems where fungi can provide insights. I mean, that's a long-haired answer to a simple question, but I think we have cases where we can clearly say that fungi are neglected based on current conservation schemes. But it's a little bit, it's a few days too early to really answer in more detail because we are working on a manuscript to write about this.
Okay, thanks a lot for the questions, and to Jakob. I don't know if should we wait some minutes if someone will still have some questions?
No, I
or
think we are
Yes.
over time. I think it's fair
Yes.
to say that was the last question, and of course you're all welcome to send an email or contact me by other means. But I prefer emails if upcoming questions are rising.
And also, I'm pretty sure this year there will be more papers published within FunDive, so maybe some questions will be answered in this more like, let's say, professional way with the scientific paper in hand. Okay, then should we, Glenn, close the first lecture for today? And yeah.
Yeah, I think so. So let me thank Jakob one last time. I think he really presented well that FunDive is a beast with many heads, and that we're doing a lot of things, but somehow they all end up with the same goal of putting fungi on the conservation map. We got a lot of questions, which is really nice. We got good engagement. Not all questions related to FunDive, but it shows that it's a broader thing, right? These things, and we only not draw on Jakob as his role as a general leader of the FunDive package, but as his general knowledge and as a mycologist. So with that, I would like to thank you all and hopefully see you next month. Thank you.
Thank you very much. Thank you.